So good afternoon. It's been really fun hearing all the talks so far today. And I'm really happy to now tell you a little bit about where our project stands. So our project is to create a human genomics analysis interface for Facebase in order to leverage a lot of the human genomics that are out there, but that are difficult for a lot of users to gain access to. So our project scientific team includes me as the PI, Eleanor Feingold, Harry Hockheiser, and Elizabeth Leslie. So we have 3 goals for this project. The first, again, is to develop a software interface to allow exploration of human craniofacial genomics databases. Another goal, to identify pertinent genomics data that we can incorporate for use with this tool. And finally, to create analysis results data sets to make available on Facebase. And we'll talk a little bit more about each of these. But a very important note about this project is that individual level data will not be distributed. This is a project to explore results within these genomics databases. So for this year so far, we've been focusing on those first couple of goals, developing a software interface, and then identifying data sets to include. So we want to be able to apply some of the very typical human genomics analysis software, for example, Plink, to human genomics data that's available, in most cases publicly available through dbGaP or other manners, with access to these analysis tools through the hub. So far we have set this up for our development and testing phases, where we have our raw data for these analyses located on a secure location at Pitt. We'll be talking to the hub in the near future about what the best long-term structure is for the program. But we can certainly continue developing parts of it at Pitt, have parts of it on the hub, or vice versa, or whatever. So none of what I'm going to tell you about the development we've done so far is set in stone, but it's to show you what we're envisioning for these tools. So really, some of the main purposes of these are to allow Facebase users to explore genomics data or ask initial research questions without having to go through the laborious process of downloading the individual level data. Then, after they've explored the data, they may then decide to download the individual data, but this gives them a little bit of a playground to see if they're even interested. So one of the first steps at the Pittsburgh side was to develop a secure network infrastructure to do our development in, and it's pretty typical. We have one of our computer clusters. We've carved out space for this project. So we're storing raw data on the head node. We have about 16 terabytes set aside at the moment, and that's in a secure FSMA zone at our network operating center. Then the web server is where the access tool and the results databases will be residing, with access through the internet through the hub, say, if we stick with this kind of organization. But it could end up totally at the hub, depending on our subsequent conversations. Just what I already said. Okay, so the other things we've been doing so far is to develop a draft web page for the interface, implement P-Link or Plink, which is one of the most common human genomics data analysis programs these days. We have other packages in mind to implement later, especially as we're looking at things like sequencing data. P-Link is really great for SNP data. You can do certain things with sequencing data, but there's other packages that we're hoping to do for some of the sequencing data. We started formatting available genomics data to be implemented in the tool, and we started with the Beatty GWAS data set for development and testing. Created results databases from that raw data using P-Link, and then developed some preset displays of results with a longer-term goal of also having custom calculations in place. So now I will turn to the demo. Okay, so the homepage just says a little bit about the project, just has a picture to brighten it up a little bit. Then on the projects button will be a list of the projects for which there's data available. So right now, we just have this Beatty project set up. So there's the title of the project, the dbGaP accession, study accession number, and we'll eventually have a link directly from here to dbGaP if you want to download the data. A brief summary of the genotyping, the panel that was done, and the number of SNPs. Now, this is the type of thing that before we finalize how we're going to set up the tool, we need discussions with the hub on how to index or use metadata. The Illumina panel would be analogous to an instrument, or the number of SNPs could be, again, another type of metadata. And then a little information about the investigators. Then if you click on the study title, there's greater amounts of information about the project. Again, some of the basics, a little more of a description. Then right now, there's just a verbal description of the sample. Something that we will add is an additional button to look at the composition of the data set in greater detail. This happens to be trios. There's 1,000 Caucasian trios, 1,000 Asian trios. So that type of more detailed information would be at a separate page. In addition, we would have separate pages about how the data was analyzed, which phenotypes were used, if the principal components of ethnicity, for example, were used for particular analyses, and so on. There's a publication list for publications that have resulted from use of this data, and clicking on the pub sends you to PubMed. Then there are two options for working with this data. Right now, we've set up some examples under this Explore Project Results. And this is some preset visualization of project results. Then, although we haven't set this up yet, there will be a custom calculator where users could do their own calculation that they're interested in. For example, if they're interested in a particular SNP or a particular gene or a particular pathway, we would allow them to specify what they would like to come up with. So here's an example, again, of what this Explore the Project Results for the preset results looks like. You can choose the study you want to include. Right now, we just have the Beatty study. These will be very much customized according to the particular project, but in this case, we have Asians and Caucasians, so you can pick Asians. You can pick a Manhattan plot for all chromosomes or for any individual chromosome. So Manhattan plots are minus log 10 p-values of the results at every SNP in the SNP panel, and they're arranged by chromosome from 1 to 22. You can see we have a number of nice peaks on here. Anything over a value of 6 is genome-wide significant. So this is a very typical visualization for human genotype data. You can switch to the Europeans, so you can sort of compare at a glance what the Europeans and Asians look like, look at the overall values. You can look at just a single chromosome, again, switching between the populations if you wish. You can click on this and then save it if you just want to save that one image. Oops. There's also options to download all the Manhattan plots for the Asians, all of them for the Europeans, all for the combined data. So among the other things that still need to be added are the very important attribution information that we wish to include both for the specific project and for Facebase. And that type of attribution information would be added to all the visualizations that come out. Locus zoom is another very typical way to visualize data. And we set up some presets for the top hits in the Beatty data. So looking at 1p22, for example, over the whole data. This is, again, a minus log base 10 p-value representation of the data, but now it's zoomed in to a specific peak in the overall results. So it gives you the genes, their start and end points, their exons and introns, and again provides the p-values, and up here the most significant SNP in that region. Just like the Manhattan plots, you can toggle between. Or you can ask for download of all the locusomes. Again, the final thing is the custom calculations coming. Okay, now I can just go back to my PowerPoint if it allows me to do that. We have to get this off.
Just start up that.
I think, yeah.
Oh.
Let me try.
I'll let you do that.
Yeah, I think it's just this part.
Okay.
Okay.
Okay. So in terms of identifying analysis data sets, as I said, there was the Beatty project, which you saw the GWAS results from. There's also sequencing from the Beatty group that was part of Facebase 1 and will be building some things to look at that sequence data. There's other data sets that are in dbGaP, like this Guatemalan GWAS of some of our OFC data. Then there's two particular projects from Rich Fritz and our group looking at facial variation in Tanzanians and facial variation in Caucasians, where there's also GWAS data available. So we have the GWAS data in hand for Rich's project, and the project for the Caucasians is slated to have the genotyping back in about a month. So a couple things about these two projects. The phenotypic data was part of the Facebase 1 project, so that's the data we've been just talking about with the consent issues, the facial measurements and facial images. So that data is actually in Facebase, but the genomics data will be in dbGaP. So this can be a nice opportunity for someone who's exploring that data on the Facebase hub to have the ability to go directly to this tool to also look at what some of the genetics results are for that data. There's a very large targeted sequencing project that we've recently completed that we hope to make part of this tool. And there's also a very large GWAS that we're in the last stages of cleaning that we'll hope to make available through this. So things that we still plan to do this year are continue to refine the analysis tool with a lot of collaboration with the hub, getting in the acknowledgments, the provenance, analysis details, links to dbGaP, cross-linking with other Facebase projects. So ultimately, I'd love to see some way for people who might be looking at the mice data and say, wow, this is an interesting gene. I wonder what it looks like in human clefting. Then they could go right to the tool, bring up some of the data sets and see, oh, there's no association, or yes, there is association. Let's be very excited about that result. We'll start implementing some of the custom calculations, allowing users to specify more with greater granularity what they wish to look at, and then discussing with the hub how to annotate and tag these data sets, both not the raw data sets, but the results data sets perhaps, or the image data sets that we're creating, these preset images or images that users who come to the site create. So we'll be working with the hub, and there's also some issues for both the hub and the steering committee to think about. For a project like this, what should our design, annotation, and metadata principles be before we finalize how we're going to set this tool up? How will it be implemented? I mean, is it best to have the gateway on the hub, but keep the data and the analysis software at PIT? As I mentioned, cross-linking with other Facebase projects, both the facial norms and the Tanzania sets, but also the animal projects who wish to cross-link their results with the humans, as integration is a big theme of Facebase 2. In terms of the data itself, we have some of the embargo issues that people have been talking about with data sharing for quite a while now. And a number of this data is quite new, so there will be embargoes on having so the embargo really is on publishing on data, but we probably should consider following the embargo dates in terms of having these analysis tools available on this data. As far as user access to the tool, I would suggest that we consider having it for registered users. But is that a good idea or not? So again, something to think about as a steering committee. And that's some of the high-level points I wanted to bring up. Happy to answer questions or entertain suggestions. Steve.
Hi, Mary. Why would you want to keep the use of the tool for people who are registered on Facebase only?
So one of the things that I would like to do with the tool eventually is to have a sandbox set up for users so they could save their sessions, and that once they get a result, it would feed back into the results databases of the tool. So if they've already calculated something, it's something, basically a preset that someone else could pull up. And I think that, I could be wrong, but I think that would be facilitated by being registered as a user. Yeah, I don't know. Yes.
At the beginning of your demo, you said something I'm guessing you didn't actually mean to say, but I just want to.
Uh-oh.
So you were showing a link to dbGaP, and you said that people could click here and download dbGaP. That I assume you just mean that's just.
I just mean that's where to find it in dbGaP.
Okay.
And then they would still have to go through the whole dbGaP process. Absolutely. Yes.
Is genome browser visualization of this data in the works? I mean, ideally, for example, I would love to be able to pull a log to see our epigenomic data access and then see SNPs falling within, certain enhancer regions that we're mapping.
Right.
And
so
if you could summarize the question.
Oh, excuse me. So Joanna is asking if there are any plans in the works to work with the genome browser to include some of the other tracks that are being developed by Facebase and others, along with this human SNP genotyping. And I didn't have it on one of the slides, but yeah, that is one of the goals near the end of this project. Again, as part of the integrative activities with the other Facebase projects, having some of these results databases be tracks on the browser that we could then pull in the other Facebase tracks and look at all the data simultaneously. Yes. Emily.
Mary, about the embargo, I don't think actually there's any more publication embargoes, and NIH has moved from a publication embargo to more of a delayed release policy on the data. So then it would be, if the investigators are still allowed to have all the data to themselves for a certain period of time, it would be whether they wanted to allow it to be analyzed in Facebase before that period was up or not. It would be up to the investigator.
Yeah, these have been shifting sands, so sorry.
Right,
Pay
yeah.
attention to Emily, not to me on that point.
Can you kind of summarize what she said?
So Emily Harris from NIDCR pointed out that there's no longer a publication embargo under the new data sharing rules, but instead they're turning to a delayed release of the data. So there is a time period where the investigators are allowed to do analyses and publish, but then it's released with no restrictions to dbGaP, which would presumably be pretty much the same principle then we would follow at Facebase. Okay, thank you.